So good afternoon, everyone. My name is Alex Frangi. I'm professor at the University of Manchester. I'm the director of the UK CEiRSI, or Centre of Excellence on In-Silico Regulatory Science and Innovation, which is an initiative from the UK government that is supporting these training webinars and helping to disseminate the activity across the community. So the CEiRSI is funded and has support by Innova UK and the Medical Research Council, or MRC, and is sponsored by the Medicines and Healthcare Products Regulatory Agency, of which we are very glad for their support and collaboration. As the UK's first centre of its kind, we pioneered the use of In-Silico methods to enhance regulatory science and innovation, and our webinars will delve into the latest advances, best practices, and applications of In-Silico technologies, fostering collaboration and knowledge sharing among stakeholders to drive progress in healthcare and medical research. And to finalize as well, I want to say that this particular webinar is our third pilot of the In-Silico Regulatory Airlock pilot. And this is an initiative where we take in turn specific problems that are in a pre-competitive space, and we use them to drive learnings around different aspects. And in particular for this year, we have selected credibility assessment as one of our main focuses. And this particular session will delve into credibility assessment in the pharmaceutical domain. So without further ado, I'll hand over to my colleague Dan Scotcher, who is going to take you through this session today. Thank you, Dan.
Okay, thanks, Alex. So welcome, everyone. So my name's Dan Scotcher. So I'm a lecturer at the University of Manchester. And my expertise is in kind of PBPK modelling, and so I've been really involved in trying to help put this pilot together with the excellent team that we've got. So to begin with, I'd like to ask the rest of the pilot team to introduce themselves, and then we'll go through the agenda today. So starting with Rajat. Thank you.
Yeah. Hi, everyone. Thank you for setting this up, Dan and Alex. I'm Rajat from GSK R&D. I'm in the Clinical Pharmacology Modeling and Simulation Department. We look at model-informed strategies to enhance drug development and clinical trial success.
Thanks. Amin?
Hi, my name is Amin Rostami. I'm Professor of Systems Pharmacology at the University of Manchester, but also 50% of my time is dedicated to the biggest, basically, biosimulation firm that helps the pharma called Certara, and I'm Chief Scientific Officer there.
Okay, thanks, Sue.
Good afternoon. I'm Sue Cole, and I head up the Clinical Pharmacology Group at the MHRA. We're in the Innovative Medicines team, so any modelling that comes in the submissions, it would usually fall to us to assess. And as I said, this is focused on the new medicines themselves.
Thank you. Alex.
Hello, my name is Alexander. I'm the lead of Systems Pharmacology at SQ Labs and specializing in PBPK, QSP type of applications.
Right, we also have Rachel, who's going to be part of the panel for the Q&A. I'll ask you to introduce yourself now. Thank you.
Thanks. Hi, I'm Rachel Rose. I'm a Senior Director in Certara's QSP modelling group, and I lead the developments of our disease and vaccines platform models.
Okay, thank you very much. So as you can see, our panel represents nicely academia, industry, and the regulatory space, and software platform providers. So we have good representation from the different areas here. Before we go further, I'd also like to just thank Saima and Lara, who are both on the call, who have done an excellent job in supporting us in the technical side of these, setting up the Eventbrite and behind the scenes work. So again, thanks to those. So I know we'll run out of time with all the discussions at the end. Okay, so with that, we'll move to the agenda for today. So we're going to begin by going through the aims of the pilot, what we're trying to achieve, addressing a question of interest, and start with some kind of higher level things around what QSP models are, how they're used in vaccine development, and address context of use, before moving on to more regulatory focused aspects, and then we go into the real details of various frameworks and approaches that's been proposed for using QSP and other modalities that may be applicable to QSP, and provide a critical analysis of these. Okay, so the aims of the pilot session today are to establish requirements for credibility of computational modelling and simulation in regulating vaccines. So vaccines is really the focus in terms of therapeutic area, although many of the things we'll discuss I'm sure will be applicable to other areas, but it really helps us to narrow our discussions in terms of providing some critical input to focus on vaccines today. We're looking at model-informed evidence in regulatory decision-making, and as you'll see, we've again selected a particular question of interest and context of use with its own model risk to help us have some productive discussions, but it's really envisaged that this is the starting point and feeds into the wider discussion around using model-informed evidence in regulatory decision-making. So we narrow the pilot to be productive, but of course we want to generalise later on. We've identified a number of frameworks that we'll address today to look at credibility of QSP models and align them in terms of regulatory standards and guidance that we'll go through, and also propose strategies for demonstrating model credibility. And we think about really, as you'll see, some of the specific nuances of QSP models compared to other modelling modalities. Okay, so to begin with, let's just give some context of how vaccines work. So the idea of a vaccine is to trigger the immune response, and there's various ways that this can be done. But in the effect that we're looking at, the graph on the right-hand side shows that after your first vaccine, the antibody, which is showing your immune response, will have a small increase and then go back down. So then we need to have a second vaccination, which builds on that primary immune response to really have a much larger secondary immune response. And this secondary response is larger than the primary response and lasts for longer. This is where, when we had, for example, the COVID vaccines, you would have your first vaccine and the booster, and this is very common. And so, as we'll come into this timing of the first and second vaccination will become relevant later on. So with that, I'd like to now hand over to Amin. Oh, sorry, no, I've got one more slide, sorry. So the question of interest for this pilot is to look at this optimal dose schedule and boosting interval between the first and second vaccine and think about the optimal vaccine dose. I'll let Rajat explain some of the details of what we really mean about that. Of course, there are plenty of other questions that circulate around this. We're really interested in how do we optimize immune responses in terms of this antibody response generated by the vaccine. And now I will hand over to Amin to talk about benefits of MIDD. Amin.
Thank you very much, Dan. I think, Dan made it, intentionally, too simple to start with. And, what, he gave with regard to the vaccine, you can start to imagine that there are many decision points, to get to that particular graphics that you have seen. For instance, what the dose should be in the first instance versus the second instance. What the time lag between the first instance and the second instance. And whether it is going to be different for different kind of people, etc. So there are many decisions involved, and the modeling basically comes to to help there. One of the things that, the model-informed drug development, for many years people have been questioning, and I have to confirm that this is not necessarily replacing actual clinical studies, but to accelerate, select them, properly, and whenever they are not possible, to have better decisions on the basis of these models, as opposed to what we call, sort of ad hoc decisions that they are not based on, any evidence. And The other challenge has been to prove there actually the value. And this particular slide that you are seeing, it is referring to a recent publication by colleagues from Pfizer, although the similar kind of thing is now available from many of the other companies that some of them publish, some of them they talked in conferences but not published. But it is actually indicating the exact value, added value that these are bringing, particularly with regard to direct saving, with regard to the money that you can go and investigate yourself. But there are also elements that they are bringing up that they haven't put intentionally any monetary value, because, you can actually debate, how much that saving would be by 10 months earlier bringing something into market. Imagine that, you are talking about the COVID vaccine, and of course, whoever comes to the market earlier, if the monetary element, is calculated, would be massive compared to someone who comes, much later, and so on. And then many of these, they are, going to be shown in the example that we have selected for you, and intentionally we picked this from what actually Dr. Dezikan, and the team, they did, because it demonstrates the value and practicality of the use. And of course, we will talk about the challenges from the regulatory side, that they want, everything to be a fair play, and the impact of that. Can we move to the next one, Dan, please? So I am coming from a corner of the QSP, which is known for the people who are not in the area, is as PBPK, physiologically based pharmacokinetics. But the PBPK and the other side of QSP, they are basically looking into two different elements of the drug's action. One is that what the body is doing to the drug, or the external element that we are putting in, and the other one is putting what, once the drug is there, what it is doing to the system itself. The key element of all of these new models that we call systems-based models, they are on the separation of the drug element from the system parameters. So you can consider, in the case of vaccines, we are talking about immune system as the system. It is not necessarily, drug, dependent. Immune system is working its own way. And the separation, has helped us to achieve many, many different things, including translation from in vitro systems, and also from animal to human whenever that animal, data are actually relevant. However, this particular, publication that, I was asked in 2012, to put into Clinical Pharmacology and Therapeutics, which is the highest research journal in the field of clinical pharmacology. If you click, the other key button, actually, when they asked me to write this, I was very proud, that, they have asked, this, and we are putting that in, etc. And it went to an element that was called state of art. And you get even, double, I would say, happy that, it is a state of art. That means, that's in the cutting edge, etc. But if you press the other button, if you think about it, that is basically saying that at the time, if it is a state of art, not everybody accepts it. So imagine that, you are going to Tate Gallery or New York Art Gallery. There will be elements there that not everybody, can agree whether this is art or not. And it takes a long time before basically everybody settles and say, this is really, really good. And therefore, in this particular article, by putting it in the state of art, they were telling us that it hasn't come to a point that everybody agrees on. And the situation has now changed. Please, move to the next one. So with the recent, I believe, if you press, the other button as well, I think everybody in the pharma space, at least, if they are not, in the medical space and so on, they are familiar with the fact that FDA announced that they are going to get rid of, many of the animal studies in particular space related to development of these biologics and monoclonal antibodies. But this is a massive, massive change that now we are moving from the system that we used to have, in the book title you can see, one for men, two for horses, just on the basis of the size, or what we call allometry. And we are going into that, how the details of these systems, that they are actually different from each other, and they are affecting, therefore, the decisions that we are making for the adjustment, etc. And the same applies when we are going from, for instance, adult to pediatric, as you are seeing there, and also within different, genotypes, phenotypes, ethnic groups, etc., etc. If you go to the next one, please. And I think I'm handing over now with this to you, Dan, back.
Thanks, Simon. So we're next going to move into kind of what the drug development looks like for vaccines, what a QSP model is, and how drug modelling and in-silico evidence can help that. So Rajat, over to you. Thank you very much.
Yeah, thank you, Dan and Amin, for this wonderful introduction. So I wanted to start out with a broad overview of what the vaccine development pathway looks like in terms of both time as well as the key questions and decisions that are to be taken at every step to get a molecule from the lab to the clinic. So the vaccine development is broadly split into 4 phases. The first is drug discovery, where we look at how vaccines against a particular pathogen can be developed. What are the optimal routes that the vaccine should take? For example, should it be an mRNA-based vaccine or some other modality, and so on? And if there are other vaccines that are already available, and what we can learn from them in order to create a more effective vaccine. The second phase is preclinical, where some of the candidates that pass the discovery phase are tested within animal models. And this is where, as Amin was mentioning, there is a lot of scope to phase out or compress this phase and replace it with model-informed simulations that could directly enable the molecule to get into clinical trials. The rate-limiting step of any drug or vaccine development are really clinical trials, where initially the vaccine is tested in a small clinical trial with a limited number of people, mainly to look at safety profiles and so on. And then we transition to larger phase 2 and phase 3 trials, which can take a lot of time because we actually gather information about the vaccine's efficacy. So we look at vaccinated versus unvaccinated populations, and we confirm that vaccination actually protects people against infection and disease. This is then followed by approval, where the entire package of data is submitted to regulatory agencies such as the US FDA, the UK MHRA, the EMA, and so on across different countries. And then hopefully the respective agencies approve the vaccine for deployment within their respective spaces. And phase 4 essentially comes from real-world studies and follow-ups, which can lead to tuning of the variables that are involved within the vaccine. For example, we might decide that some of the individuals might need higher doses, such as elderly or immunocompromised and so on, whereas some of the other populations, such as pregnant women or pediatric populations, may require a different dosing schedule. So all of this comprise vaccine development, and you can see this is a decade-long process for most vaccines, and takes a lot of money. Which is why we are really looking for model-informed approaches to compress the timelines as well as reduce the risk of taking the wrong decision across the phases. Next slide, Dan. So here is how we think about model-informed approaches integrating with vaccine development. A good analogy for this is weather forecasting. So if we want to step outside of our house tomorrow, we would like to know whether it's going to rain or not, so that we can either carry an umbrella or take the right contingency measures. Models do exactly that. They model the actual phenomena or clinical trial or experiment that we are planning to conduct in the future, and they make predictions about how that experiment is going to go, so that we can narrow in on the most optimal set of variables that is likely to enhance the probability of success. An important thing here is to integrate models through the development continuum, so that at every phase, when new data is generated, that is fed back into the model, making them more and more accurate, so that the confidence in vaccine as we progress through development increases and the uncertainty within the model drops. which is shown below. Next slide, please, Dan. Just to give you an idea of the sort of decision points that are involved at various levels. On the left is more of the discovery end of the spectrum, and on the right is more of the real-world deployment end of the spectrum. As I mentioned, initially, the decisions could be around the optimal modality, the number of antigens. For example, we know that a lot of pathogens have multiple serotypes. Pneumococcal bacteria, for example, have over 70 serotypes, and we cannot vaccinate against all of them. So we might integrate epidemiological data to figure out which are the most important serotypes causing debilitating disease and hospitalization and narrow on those. As we progress into preclinical phases, the idea is to use the data from these animal models to figure out the optimal dose and dosing schedule, and we'll talk about this in a little while. Whereas as we proceed through late clinical development and so on, it's really about the safety profiles, the robustness of immune responses, and how long immunity lasts. You might know that different vaccines have different levels of durability. For example, we get a measles shot once in our lifetime as babies, and we more or less have lifelong protection, whereas with respiratory infections like flu and COVID and so on, we need to get repeated booster shots probably every year. And real-world deployment could be in the setting of a pandemic versus a more normal setting. How should vaccines be deployed so that we combat the infection most optimally? Next slide, Dan. So the kind of models that are typically deployed within vaccines fall under this umbrella called as QSP. It's an acronym for Quantitative Systems Pharmacology. As Amin mentioned, this is a confluence of multiple sub-modeling disciplines, one of which is physiological modeling, where we actually model the pathways and the biological interactions which are within the immune systems. And these are wedded to models of pharmacology, where we look at how differences in vaccine doses and the interval between doses actually perturbs these biological pathways so that we get the optimal response that we want. These sort of models are great for forecasting because they are built on previous knowledge of human immunology. We have accumulated a lot of knowledge about human physiology so far. And therefore, with the right amount of tailoring to cater for our particular vaccine, we can forecast how the responses would look like with a certain drug or dosing regimen. Next slide, please, Dan. Now, as you might imagine, this sort of methodology of modeling can be quite complex. And many different classes of models can fall under this broader umbrella of QSP. Some of them are shown here from a survey within the pharmaceutical industry. As you can see on the right, this can go from mechanistic models of the human physiology and drug perturbations all the way down to agent-based models and machine learning approaches, which are more data-centric rather than mechanistic-based approaches. And it's likely to account for all the complexity that we observe across humans, across populations. We need a confluence of multiple methodologies to effectively drive decision-making using QSP. So the highlight of this is really that QSP is a constellation of tools that have to be utilized and deployed appropriately. Next slide, Dan. Now we'll focus on concrete examples where we show how QSP can influence and inform vaccine development. If I can take you all back to the initial days of the pandemic, we saw that vaccine development occurred at lightning speed, at an unprecedented scale of months rather than years and decades. And vaccines were approved within 2020 for deployment across populations. Now, a key consideration was that the clinical trials with the vaccines were conducted with 3 or 4 week intervals between the first and the second dose. Whereas when they were deployed across various countries, I think Israel was the first country where vaccines were deployed, with UK being the second. The public health organizations decided to deploy the vaccination with a 12-week interval between the first and second doses to ensure higher first dose coverage. So this sort of sparked off a discussion among immunologists and clinicians as to whether increasing the interval between the first and the second dose would possibly lead to a reduction in vaccine responses or not. So at that point of time, within our group at CEiRSI looked at In-Silico trial simulations, which were calibrated based on actual data from Pfizer and Moderna vaccines. And what we actually found out was that increasing the prime boost intervals from 3 weeks, which is the orange line that you see on the left, to 15 weeks, which is the sort of the pink line that you see on the right, actually leads to an increased immune response, with about 7 or 8 weeks leading to optimal levels of antibodies. So more importantly, we showed that the 12-week interval is actually superior to a 3 or 4 week interval in terms of levels of antibodies that were raised. So this result, if you could press once done, these results, after we made the predictions in December 2020, were confirmed in a real-world study within Oxford and gave us a lot of confidence that the deployment was not going to impact vaccine efficacy. So this is how we can use virtual trials to forecast complex scenarios which might occur in the real world. Next slide, please, Dan. This is getting into the nuts and bolts of the virtual trials a little bit. You might be wondering how we actually calibrated the model and so on. So on the left, you can see the colored lines. Each of these 9 colored lines correspond to actual patients who were enrolled within a Moderna phase 1 clinical trial. And we calibrated our vaccine QSP model to conform to the range of antibody responses that we saw in this real clinical trial. And the QSP model enabled the generation of virtual patients. These are patients on our computer with whom we can run a whole lot of what-if scenarios to predict and inform decision-making, like we saw with different time boost intervals and so on. So each black line you see on the left is a virtual patient, and the collection of virtual patients is much larger than the actual number of patients enrolled in the phase 1 trial, which gives us a good measure of heterogeneity across people. At that point of time, one of the key questions with COVID-19 is, when should we boost, or how long does the immune protection last? Again, remember, this was 2020 when a lot of data was not available regarding vaccine responses and immune durability. And we predicted, as you can see with the graph in the middle, that the antibody responses are likely to decline below the protection threshold, which is the horizontal red line, between 300 and 600 days, which means that we'll probably need a booster 1 to 2 years, which is exactly what the real-world data showed us later on. And this is, again, another example of how we can forecast complex real-world scenarios way before the actual scenarios happen, and therefore optimize vaccine development. Next slide, please, Dan. Next slide, please. The other way we can use In-Silico trials is to optimize dosing across various subpopulations. For example, we know that young and old adults do not respond the same to different vaccines. The flu vaccine, for example, for older adults has 4 times the dose as the vaccine for younger adults and so on. We wanted to know if this was the case for COVID-19 vaccine as well. And within our virtual clinical trial framework, we could test how different doses of the COVID-19 vaccines would function within individuals. So here on the graph, the x-axis is time. So this tells us about the durability of immune responses at different dose levels. On the y-axis are the percentage of responders. These are the fraction of our virtual patients who go above the red protective threshold that we saw in the previous slide. And for example, 100% means that all the individuals who received the vaccine elicit antibody titers above the threshold required for protection against disease. So these results were quite interesting. So if we look at the younger adults on the left-hand side, we see that with 10, 30, and 100 micrograms, all the individuals initially achieve 100% of response, but the waning of immune responses is actually quicker with lower doses than with higher doses, indicating that higher doses may enhance durability of responses. In contrast, with older adults, with the lowest dose of 10 micrograms, you can see that we never reach above 75% of protection, which means that 25% of individuals who receive the vaccine may not generate antibody responses above the protection threshold. And therefore, this means that if we want to pick a vaccine dose that would work across young and old adults, we would probably be better off either picking a 30 or a 100 microgram dose. And these sort of simulations are constantly used by vaccine makers to inform decisions about their dose and intervals. Because at the end of the day, having a single dose level is much more easier for manufacturing and supply across the globe. Next slide, Dan. Lastly, I want to emphasize how we can use something called as digital twins to enhance vaccine development. So digital twins are virtual patients, but they have a one-to-one mapping with real patients involved in a clinical trial. And these sort of digital twins are very helpful for what-if scenarios and dose selection. One of the issues with phase 1 clinical trials are the low sample sizes across the dosing arms. These are clinical trials where typically a range of vaccine doses are tested, and that optimal vaccine dose is chosen, but the low sample size can lead to a lot of noise in immune responses. So the concept of a digital twin is illustrated on the left, where the blue circles are actual clinical data, and the digital twin is the purple line that passes through the points. This is a made-up example. So we assume that, let's say we have a phase 1 trial where we have 7 dosing levels from low to high, and they all have a lowish sample size of between 13 and 30 or 29. So if we just take the antibody responses from the second dosing level, you can see that they are plotted below, and there is a lot of heterogeneity across individuals, with some people developing very high antibody levels and some people developing very low antibody levels. So how do we correct for the sample size and estimate the true measure of optimal dose response? One thing we could do is use the digital twins across all the 7 dosing arms and enroll them in a clinical trial simulation where they all receive the same dose. So a digital twin, for example, from the D7 dosing arm could also receive a D2 arm within simulations on our computer. And this immediately bumps the statistical sample size, for example, with the D2 arm with 15 patients in the real clinical trial to 166 digital twins within our clinical trial simulation. And you can immediately see that we capture a much broader range of heterogeneity of antibody responses, as shown below, where each line is a digital twin, and the blue lines are actually the clinical data from the second dosing arm over-plotted on top of them. Next slide, Dan. Now, this allows us to cleanly capture a dose response curve, where we can test an apple-to-apple comparison by only changing dose as a parameter across the entire cohort of digital twins. So the underlying immune pathways remain the same. It's only the dose that changes, and this gives us a smoother monotonic sort of curve that you can see in gray, as opposed to clinical data, which might have a lot of noise. And therefore, this helps us to identify what an optimal dose is likely to look like. Could you press this once more? So here, the optimal dose, for instance, could be the dosing arm 4, which lies above a certain immunogenicity threshold that was pre-decided. But if you look at the actual clinical data, which is the red triangle, that actually lies below the dashed line. So that's how modeling can sort of integrate all the data that we have and drive more robust decision making. Next slide, Dan. So I'll skip this in the interest of time. The other way to use digital twins is to shorten clinical trials. As you know, a lot of clinical trials go on for many years, where we look at immune durability and so on. For example, there are trials with Shingrix vaccine and so on that have gone on for a decade, where we look at the slow loss of protection over time. So one thing that could be done very early within a clinical trial is to build digital twins and then use digital twins to forecast how long the immune responses last. The process typically goes like this. Let us say that a clinical trial has proceeded for a certain amount of time, and we have 3 data points. We build a digital twin to it. Can you press next, Dan? And using the digital twin, we make a prediction for the next time point where the data is collected. Can you please press next? So here we could have three outcomes. The first outcome, which is very unlikely, is that we have a crystal ball and that the new measurement which is taken later on exactly corresponds to what the model predicted. Once more, Dan. The more unfortunately encountered outcome is that the model did not capture the new observation as well. The outcome that we are looking for is that the model captures the later time point within an acceptable threshold of error. If this is the case, we accept the model and we use the data from the new time point to train the model, and then we can forecast the next step. So typically the model converges to a pretty high level of accuracy within a few cycles of this train and confirm cycle, and then we can use it to robustly extrapolate to much longer time scales, which might compress this decade-long clinical trials into more of a 3 or 4 years long clinical trial, thus resulting in significant savings in time and money. Dan, how much time do we have? If we are out of time, I'm happy to skip over the slide and give it to the next section.
No, go ahead, Raja. I'll see if we can cover the context, which is quite important for this particular pilot. Thanks. Yeah, go ahead.
Thank you so much, Dan. So this is probably the last case study that we'll be considering, and this is how to optimally design clinical trials so that we inform the next phase of vaccine development and therefore, de-risk the entire vaccine development process. For example, let's say that we are designing a phase 2 clinical study with 3 dosing arms and a placebo. So if you look at the graph on the left, the 3 dosing arms shown in red all elicit similar antibody responses, and the placebo, of course, has less antibody responses compared to the dosing arms. One could argue that this is actually a failed clinical trial because we cannot elucidate a dose-response curve from this. And as shown with the yellow, green, or blue lines, there are multiple dose-response curves that can all explain the 4 dosing arms that we see. One of the ways to de-risk this is to use models to already predict what a dose-response curve could look like, integrating all the previous information that we have about that particular disease and preclinical data and so on. And then we could say that, look, here we are missing a dose in the intermediate regime, which is yellow, so it would help to test that dose. And why this is important is consider scenario 1, where a dose tested in this region has a lower response compared to the other 3 doses. This helps us narrow down the dose response as the green curve. In contrast, if we have scenario number 2, which is that the lower dose tested is equivalent to the other 3 doses tested, this still, of course, has some uncertainty, but much lower than the previous case. And we could think about using QSP models to distribute the patients across dosing arms so that the overall sample size is the same. So the amount of resource commitments are the same essentially, but we can drive better decision making from the clinic by optimizing the clinical trial designs. Thank you, Dan. Over to you and the next section.
Yeah, thank you very much, Raja. That's really, really great and really useful to contextualise what's coming next in terms of the regulatory part. So to begin that, over to Amin. So Amin's just going to give brief highlights of where things are in terms of the use of QSP models in the regulatory space.
Thanks, Stefan, and thanks, Rajata. I think, the biggest element that perhaps now I think the people who are not in the pharma space start to realize is that the QSP, not from us, in academia or the pharma users, but from our regulatory colleagues, is seen as the post-hype in the hope-hype cycle. And I invite, people to have a look at this particular piece written by Dr. Issam Zeine, who's the director of Office of Clinical Pharmacology at FDA, a few years ago he published that, and despite the fact that, I will argue that PBPK is ahead of the rest of QSP in this curve, but, I think at least, we agree that definitely we have passed the hype element that, you may, sort of, be familiar with, that, we had 20 years ago when, all these models started with, with the initiation of companies like Entolos and so on. So I think everybody agrees that we have passed it and we have got practical examples. If you go to the next one, and those practical examples, they are not just, sort of, as Rajat was saying, are they now in the internal decisions, but they are going into many other areas that they are accepted actually by regulators, and colleagues from regulatory space, obviously, they will talk about some elements of those. But this is another example that takes, you through some of the complexities that, these models can answer, which cannot be answered, at least easily, by doing clinical studies. And here, without going into any of the abbreviations and, the names that you are seeing there, it's in fact, I simplify in the form of, we had a new entity, drug entity, which is what we call bispecific, sort of, that was hitting two targets at the same time, that is already in the market and it's showing, some effectiveness. And the next issue is that, somebody brings up a new entity that by hitting three different targets at the same time can bring additional benefits. Now the issue is that how do you adjust this new modality in a way that it hits target 1, 2, 3 with the different affinities and so on. And you can have enormous number of permutations that you have to test before getting into actually to that point. But one, as Rajat was saying, that making the decision and going quick and making sure that you have actually taken the optimal decision makes a huge impact. And this story is in the paper that you are seeing at the bottom part. So these are actually making their way, as I said, now to the regulatory space, and they are filling some of those gaps that we cannot answer with the clinical studies. If you move to the next one. So this is again another report by FDA colleagues that recently they have published, and they, without going into the details, you can clearly see from the left-hand side that the increase in the number of the QSP models submitted to FDA, according to this report that comes from FDA, is certainly supralinear. It is, it's not logarithmic, it is definitely, is going much more than what you expect from a linear growth. And if you look at the areas that they are actually addressing, it is the biggest space of it is deciding on what dose to give in a way that it is effective, but at the same time it is avoiding of any side effects. We can't, completely avoid side effects, but basically optimizing these, sort of using these models at a stage that we call in the drug development phase 2, decide on the dose that's going. And many of the actually entities that are failing, and in the early preclinical studies, the models that people thought that they are going to be useful, later on they actually fail in this particular stage. And therefore this is very important stage in the drug development that these models can help us. If you move to the next one, please. I think I hand it back to you in this stage, and I will come back and later on, sort of tell the rest of the story. We have got the similar kind of thing in the PBPK, and now the numbers that they are accepted with regard to what goes into the label has come to more than, 130 cases.
Okay, great. That's great. Thank you very much, Amin. So that's kind of nicely set the scene, or kind of what we're aiming to do with the pilot is now think about the credibility assessment. So as we get into this, we're going to look across different areas of guidelines, proposed frameworks, and you've heard some of the nuances around the QSP models. You heard from Rajat about the digital twin aspect and others, and also the different variety of models that we have for QSP, where they can have just maybe 10 or 20 parameters up to hundreds of parameters. They could be dedicated models or platform level models taking long times to develop. So with that in mind, we've looked at a number of elements, and so to kick off with this, so I just, if everyone could mute if you're speaking, please. And I'll hand over to Sue to go through some of the regulatory frameworks.
Thank you, Dan. Could I have the next slide, please? I'll now put my camera on. So I get to talk about the regulatory guidance. We usually have a lot of guidance. And as Dan mentioned, there's the guidance around the use of the modelling itself, but also guidance around the question, what are we using the model for? So in this case, it's around the development of vaccines. So we're looking at the guidance that is available on the development of vaccines, and there is this quite recent guidance from the EMA in 2023, and we're looking at the guidance on clinical. There will also be guidance on the quality aspects as well. So, and in this guidance, it talks about using immunogenicity data to identify the number of doses, and also in terms of like the intervals between doses. And it also talks about that different schedules may be needed in different populations. What we have noticed is that there is no mention of modelling and simulation or of QSP in this guidance itself. And I have to say this reflects my own sort of experience at the MHRA. So when we have had vaccines in for approval, it's been really based on the efficacy data, so whether it prevents the disease, but also on the immunogenicity data. So it's really, ClinPharm involvement has really been minimal. It's really been performed by the clinical and the statistical assessors. We did see the model, the QSP model that Rajat introduced, but as he mentioned, that was really done retrospectively. The information that informed the longer interval of dosing wasn't purely based on economics. There was some clinical data that suggested, at least for one of the vaccines, a better efficacy with that longer duration. But as I've said, so we did see this model, and at the time felt that it was useful, and I think we said that it was a useful model, but it was seen as supportive, so it wasn't seen as a high risk in the whole thing and didn't undergo close regulatory sort of scrutiny. There is a lot of interest in the development of vaccines. I know at the MHRA there's interest in the antimicrobial resistance area, which is a big area at the moment in terms of the vaccines for the antibacterials, and also in terms of the cancer vaccines. And we've got a draft guidance that's out for comment at the moment. And I would say, if there are guidances out there for comment, do comment. And if you think that modelling could be useful, do suggest that. I know there is an EMA guidance out there as well at the moment on looking at vaccine development in immune-compromised individuals. So that one as well is worth looking at. I think next slide, please. So there is also guidance from the FDA. This is older, I think sort of dates back to 2011. But also in this case, there is no mention of modelling and simulation or QSP in this guidance. I think next slide. So in terms of the guidance and modelling itself, so there is a number of guidances. It's usually focused on a particular type of modelling, so population PK modelling or the PBPK modelling, and each of the different agencies have got their own guidance. So usually when you have an area where there is this sort of lack of harmonisation and where there seem to be differences, that would be when ICH steps in, so the International Conference for Harmonisation. They will pick something up and look to harmonise that guidance globally. And this is what has happened here in terms of the ICH M15 guidance. So they're looking to propose a sort of general framework that can be used in terms of the use of model-informed drug evidence in regulatory submissions and for all types of different models and globally as well. And in this guidance, they describe it as a sort of sequence of events. So we've already heard talk about the question of interest and the context of use in this particular example. But there are other things as well involved in the planning. So we look at the influence of the model, how much does this model contribute to the ultimate decision? Is there other information that's used as well? What would be the consequence of if there was a wrong decision? And that leads to this parameter, what we call the model risk. And I'll explain this a bit more in another slide. There's also consideration as well around model impact. So, and that's really the regulatory impact of the model. And this sort of consideration then goes to inform the method of model evaluation that's required. So in terms of the verification and the validation of the model. So it depends how important the model is seen to answer the question as to maybe what level of validation and verification would be required. And there's also some information in here as well around reporting the model itself and their interactions as well with regulatory. So I think that, this guidance is gaining a lot of interest. There was a page meeting, Population Approach Group Europe meeting in Greece last week. There was a whole session dedicated to this guidance. Our MHRA person is my colleague SM Kerwash. And he collated all the UK comments for this, and I know that they're working on that at the moment. If you go to the next slide. So one of the key things as well around the ICH is around getting a consensus on terminology, and I think this was really needed here. So I worked with the EMA on their PBPK guidance, and we avoided the use of the term validation. We felt that was too stringent. So in that case, you would see there's appendices that talk about the predictability of the drug model and talk about qualification of the model. Whereas FDA, we use in terms like validation. So I think this guidance is really important in terms of gaining that consensus. And I have to accept that we are going to be talking in future about the validation of the model. I think it says here that's about assessing the adequacy of the model and looking at the important underlying principles as well and comparing the model with the actual observed data that you've got. In terms of verification, I think that hasn't changed very much. I think that my feeling is that we've been largely consistent on that, and that was the same in the PBPK guidance. The verification is usually around the model code itself and the equations that are used. If you go to the next slide. So in terms of the terminology as well, we have this now thinking about the applicability of the drug model. So, what is its intended use? Previously, we might have called that whether it's fit for purpose. So, and we need to look at this as well in terms of each of this. Sometimes you have a model that might be used for a lot of different things. So you need to look at the applicability in terms of each of the questions of interest that you have. If you go to the next slide, please. So the framework that's used within M15 is not new by any means. So it's based on this risk-informed credibility assessment framework that originally came from the engineers. So this is the American Society of Mechanical Engineers, I think ASME. And they described this framework that has been adopted by M15. It was introduced, I think, back in 2019 at a PBPK meeting the FDA had by Colleen Kummel in terms of its use for PBPK models. We have seen some use of it, but I think that is going to massively increase now. So, and in this model, you're looking at, like I say, determining the question of interest and the risk of the model. And then you go into your validation and verification activities. And at the end of this, you come up with this credibility assessment that tells you, is that model credible in terms of the context of use. If you go to the next slide, and really thinking about this in terms of the example that we're thinking about today, we have this so we need to think about what is the influence of the model and what is the consequence of the model being wrong. And this is a proposal that I put. I don't think anybody else has commented on it yet. And this was just based on the data that I've seen so far. So I felt that the model influence was high because this seems to be that the decision in terms of the clinical trial and the dosing was based just on this model. It's probably not the case. I would imagine there is in vitro data and there's non-clinical data as well. And if there is, then that might drop down to medium. But then in terms of the decision consequence, I would say that was low, because in this case it's informing a clinical trial. In that clinical trial you will be looking at a number of different doses. You usually start off with the lowest dose first. And at the end of it, you've generated some clinical data itself, so then you've got the clinical data. So my feeling was that for this one, we're talking about quite a low to moderate risk, and that would then drive what is needed in terms of the verification and the validation of the model. But it would be interesting to work this through in terms of the example. I would say ICH M15 is working through now a number of examples of how their framework can be used. And I know talking to them last week, there is an interest there in looking at it in terms of a QSP model, maybe this one itself, but I'm sure that anything we did here would be of interest to them. And I think now I hand back to Dan.
Yeah, thank you very much, Sue. That was really great. So we've now covered the question of interest, context of use, and the model risk, and looked at some of the terminology around verification, validation, and so on. Now, knowing the QSP models, as we have this variety, and we also have platforms that are developed, because some of these can take 6 months or even a year to develop properly and be applied. So we have other elements that we may consider around, for example, qualification, that Amin will be talking about, and the idea of this fit for purpose versus framed for future. So once we've gone through some examples there, we'll look at the diversity of the QSP and evaluation framework for systems models, biology as a driver for validation, as well as virtual populations. And these later examples Alex will be taking us through. Okay, so with that, I'll hand back to Amin, and we'll get started with qualification and credibility.
Thank you very much. So the first piece, I'm bringing to attention of those people who are not in this space is this consortium publication with separation of the platform, which is mainly focusing on the system parameters. This is basically the common denominator for everyone who's using, and recently actually looked at, by, again, FDA colleagues, as well as, EMA, which I'm sure that you are going to talk about, versus the application of the model for particular context of use. And this separation was very, very important, because all those wordings that we are talking about, and we address that in this pharmaceutical research paper, comes on the back of those. What element we are looking at. So the qualification of, I argued in the paper that when we are talking about qualification of a medic or pharmacist or dentist, etc., that doesn't mean that their work is going to be perfect, but it is saying that it distinguishes them from somebody just on the street coming and, sort of trying to do that particular task, which obviously nobody wants, that kind of thing. But the next stage, as Sue started to talk about the validation, I think the validation issue that many people sometimes get, unfortunately, to that, sort of trap, is that if by validation we mean that definitely we need to do that particular study, clinically to show that, what actually sort of the situation is, then, modeling becomes more or less useless, because, then why we are actually using the model for? It's most of the times are the areas that we cannot address using clinical studies. And I will show you what that actually means with regard to changing the behavior with regard to actually verifying and also, long-term, use, etc. And I invite people to look at those and see the arguments on different, sort of angles and wording, etc. that Sue actually gave a very good, intro on them. If you move to the next one, I would like to actually bring an example case to clarify this. In 2012, FDA colleagues had a dilemma when it came to a drug that has been submitted to them with regard to the level of drug-drug interaction and whether that the level of drug-drug interaction in renally impaired people would be different or not. This was submitted by the J&J colleagues, and doing the conduct of such clinical study to find out whether the DDI would be different level is horrendously difficult, because not only that you have to actually, do the drug-drug interaction, you have to go and find, sort of the patients that they are renally impaired, but the alternative, now going back to the danger that, those tables that you actually seen from Sue, in many cases, people forget about what will happen if you remain silent. And this was, sort of the discussion that they had. Shall we go silent, which we know will end up with, basically what we call off-the-label usage, because, the doctors, even it is not saying anything, they will start, to do an guesswork and, ask this, that, etc. And God forbid, I don't know, Google, ChatGPT, and come up with, sort of decision that may not be the optimal one, versus doing a modeling. And in this case, they did the modeling. They used a completely different case. Therefore, it wasn't validation. It was verification of that model can answer a prior case. And that prior case was for a different drug, but the same principle, very first one in the left top corner of this slide. And they showed that they could have recovered that data and got some confidence. Once they did that and this went to the label, now they put also to the J&J that you should be monitoring the situation, and they were monitoring it themselves. And now fast forward, next slide, 10 years later, what FDA discovered was that not with a direct assessment of the DDI, but just by looking at in the market and looking at the post-marketing data, the side effects of that particular drug in patients who had renal impairment and they were getting other drugs behaving as inhibitors of PGP-N3A4, which are related to the pathway of the elimination, etc., had been statistically higher, which was an indication of what they already knew via the modeling, that this is going to raise the level much higher than other people that they don't have the renal impairment. So hence, the name of the paper, unfortunately, Joe Carrillo passed away last year, one of our colleagues who actually was involved with this. The full cycle of this took, of course, much longer. And imagine if that information was not in the label, what would have happened? Because obviously this combination effect was by the doctors who were not reading the label, because label clearly said that you have to actually monitor, there is a danger, and so on. While if we didn't have that, this incidence could have been much, much higher. Next one, please. And this brings me, to the discussion that we had on the fit for purpose. There was another piece by FDA colleagues that they have recently put with regard to many of the models that, we traditionally have done. They have been fit for purpose, and this will continue. That we have got a question and we want to answer that question. But what is changing is the behavior that we have got with regard to the models that they are only focusing on that question versus the models that they are much bigger and broader in the context that of their use, but then they are trying to answer that specific question too. And these colleagues, they are looking at the differences of the fit for purpose versus what we call dynamic models. Dynamic models because we are all the time adding the information into these models. When going forward, model is not actually, let's say, static. All the new information goes in. But the advantage that it gives to these dynamic models is that you can reuse them, and this is another term, reusability of the model, that many people in the software area, they have got that, but for the models, it is particularly in the pharma area, this was a new thing that when we brought up 2-3 years ago, but the reusability of this, sort of that it offers, it is a big, big advantage. And also, because a major part of, if you click, once more, Dan, also, when you look at, sort of how this enables the assessment in a faster way, because you are focusing on the reusable element once, but then the next group that they are using that reusable element, but they are adding other bits, you are only looking at the assessment of those other bits, and you are not redoing that. We will come back to this with this qualification of platform recently EMA has actually released, which is exactly in that line, saying that what element doesn't need to be reassessed when you are actually submitting it. I think my part is done then, and we can move now to the other elements.
Yeah, so it's a nice picture. I mean, you mentioned this, Amin, so I'll put one slide towards it just to mention that this draft qualification opinion is out for the SimSip simulator. So if you have comments on this, the comments are open, and it's worth having a look, because this is a example where there's a platform and the context of use is very clearly defined, but it enables, if good practice recommendations are used, then it supports the DDI risk for a new medical product. So the next person to use the software for this particular context of use, the predicted performance can be referenced from the qualification in regulatory submission. So it does mean that if this does go forward, the burden of providing the submit information on the qualification for the regulatory submission is reduced somewhat, although of course you would expect that the evidence around following good modelling practice or the new medicinal product would still be followed. Okay, so with this, I'd like to now bring us back towards QSP specifically, and just to remind you that we're thinking of QSPs across a wide spectrum of models that can take various numbers of parameters and different development times. And so the question that I'd like to kind of prime you when you're listening to the next set of slides is, if we have a diverse set of models that fit within the frame of QSP, do we also need a diversity of approaches for evaluating credibility? So with that, I'm going to hand to Alex to take you through the kind of current practices and then some of these evaluation frameworks. Okay, over to you, Alex.
Yes, thank you. Yeah, this was a very nice introduction from everyone, touching upon the different, let's say, terminologies and factors involved. And I think I'd like to take a little bit a step back, right, and trying to both synthesizing a few of the concepts on their higher level, and at the same time, bring up a few of those, terminological items that we've heard. And I'd like to kind of spin that around and trying to look and take a step back and try to see how we actually get to, let's say, consensus in, what needs to be done, right, for the assessment of models. We've heard that there's guidance on, in general, let's say, MIDD or model-informed drug development considerations. There's specific guidance and now evidence, and also, the qualification of PBPK frameworks coming along. But for, let's say, QSP models or QSP models that specifically target, let's say, vaccine development, right, there's still gaps in, what's our consensus pathway towards validation and qualification of those pathways. So how does that work? How does coming to, let's say, this consensus approach work? And we need to consider that this is actually not a top-down procedure. It's not someone deciding what good practice is, but it's really a grassroots type of process where you have basically the evidence of many different use cases that come to age or that are developed in our community over time. And this is just emerging, right? And then over time, right, there will be experts, there will be opinion leaders, there will be assessors, there will be people exposed to many of these models. They will start synthesizing out good practice. They will start reviewing, writing white papers, doing surveys, and then conducting reporting workshops. So over time, right, there will be iterations of synthesis of these use cases that will then basically emerge as something that can, at the very high level, basically give some guidance, right, to policymakers and competent authorities. So what we still need, however, in our community to achieve, right, is that we have this duality where we have, given the diversity of QSP that exists, right, that we have, the high-level abstraction that's done. It's a complex and long process, but at the same time that we don't lose sight for, let's say, the technical aspects of how, in practice, can this be done, right? If the guidance is too far from the practice, it's again pretty hard to apply it. And that's why we've been concentrating a little bit on this middle layer, where there's some already established, let's say, frameworks, some established overviews of what is currently good practice to give you a little bit of clustering, right, about the use cases and what starts crystallizing out as good practice. Could you switch to the next slide, please? Yes, and one of the frameworks that is being out there, that has been formulated, I'd like to focus on this one. It's been published by Brachman. And this is a very interesting framework because it has interesting duality. It both suggests a process and suggests also some technical level, let's say, atlas, right, of methodologies and techniques to be used in line with the process. So from the process, right, it pretty much follows the ASME DNP40 type of, plan, build, and assess. So right question, right model, right analysis type of framework that is also in line, right, with the ICH M15 draft guidance that's been formulated later. So it's basically you have a question-driven approach where first plan out your analysis, and then according to that question of interest, build basically your systems model and build basically the workflow, and then you pass through this predefined set of analyses until you basically do your assessment, right, of that model. And really the authors emphasize that it's more actually not a binary assessment that you do to reject or accept the model, but you have to consider these complex models as more like iterations over basically the model and its credibility in line with basically data that comes in on a regular basis. Yes, so one of the most important aspects is during the planning phase, right, that dependent on the data and dependent on the question of interest, you have to design your assessment strategy, right? And what turns out to be critical is actually the design of the data strategy. So the gold standard, of course, would be to have a prospective design where you will use external validation data to then confirm for your predictions, but in most of the cases, actually, you will be facing with basically data package and then selecting some of those data points for some holdout type of validation, for example, a dose level or one of the comparators that has not been used for calibration and basically confirming the fit for purpose of the model with that regard. In some of the cases, when for example, early clinical development, it's a relatively data poor environment, there's also examples for cross-validation. So basically doing, utilizing to a maximum the calibration data and doing n-fold, let's say, or k-fold holdouts within your calibration data and to do some cross-validation. So that can be also very useful in data poor situations, right? But what's been pretty interesting as well is that in this framework there's quite some emphasis on basically parameter exploration on top of this formal, let's say, assessment against the comparator. So that's important in that framework. So there's quite some evidence about sensitivity analysis, identifiability analysis, and later about uncertainty qualification, which is, I guess, playing a particularly important role for QSP models that inherently come with a lot of complexity, that come with a number of parameters or many parameters to estimate, so that basically, especially the identifiability and uncertainty are critical points. If you can come to the next slide, please. Yes, and that's why I think that this paper is quite useful, because it provides also an atlas of techniques, right, that you can refer to when you follow basically this process. For example, for sensitivity analysis, there's a discussion about local versus global sensitivity analysis, and also comparing different techniques, for example, derivative-based versus variance-based global sensitivity analysis. There's a discussion and comparison and actually illustration about what is a structural and practical, what's identifiability analysis, and how can we perform, what tools are available to perform structural identifiability and practical identifiability analysis. And also what's been put out in that paper is that, that there are different ways, right, and different techniques available to assess and quantify parameter uncertainty, touching upon Bayesian approaches, bootstrapping approaches, and that is also very important actually to not only know which parameters are uncertain, but that you propagate assumptions of parameter uncertainty and error assumptions basically through the model and towards basically the model prediction. Could you go to the next slide, please? Yes, the framework, however, it touches also, I'm referring to the slide before, it touches quickly about the fact that what we heard before, QSP models are rarely built for a single application in mind, and more often than not, you will actually consider different types of contexts of use, sometimes actually many contexts of use for one model. So that's a situation that's pretty unique for QSP models, and which is actually pretty hard, right, to capture in this more, sequential and linear and fit for purpose type of, qualification framework. The second aspect is that the authors already pointed to the existence of virtual populations. We've heard that this is a pretty useful concept. Existence of virtual populations, and that they can be used for calibration. But nowadays, virtual populations, they are accompanying QSP models basically naturally in the addressing the question of interest, and that has been another aspect that's been a little bit underrepresented in that paper. What I would like to continue with is basically this first point, right? So that build QSP models with different contexts of use in mind, and actually take into account or leveraging a lot of different, let's say, pieces of evidence, pieces of knowledge about the biological mechanism. And that's been a paper published by Singh et al. That basically focused on this unique positioning of mechanistic modeling and QSP modeling, which basically says that biology is really the driver. It should be the main idea, the main driver, how you actually strategize about model validation. And it's also giving you a few key limitations of how much you can actually validate given your validation data. And the picture that the authors have been putting out would be this, theoretical 3-pathway model, right? Where you would, for example, just making up an example, you would have some upstream, pro-inflammatory cytokine or pro-inflammatory pathway, then maybe some systemic, inflammatory pathway, and then, for example, also a regulatory pathway, and they would all interact to basically give you some inflammatory disease outcome. And the authors kind of pointed to the fact that dependent on where, at which places, right, you can assemble different elements for the validation or for the validation of this model, you can actually give credibility to different capabilities of the model. So they basically formulated 4 levels of validation for that model. So, for example, you could basically just qualify the type of data that you have at disposition, right? So really just using the calibration data and showing goodness of fit, that the model plausibly represents whatever you've put into the model. So there would be no basically validation of predictive behavior, but just basically pointing to a good interpolation capability of that model. The next best level would be that you go within your target, right? You would basically say, okay, I have data on other situation that targets A, and I would look into how well actually the model would have been predictive if I left out data on that other context on A. And this can be, for example, a different dosing regimen, or potentially just very similar drug that would be targeting the same, or would be targeting the same target and using the same pathway. Then the next level of validation would be that within target within pathway validation, where you would, for example, have also on top of your data for A, you would also have some data for other, let's say, targets within the same pathway. Let's say you have some upstream cytokine blockage and maybe some downstream, let's say, cellular blocker or something. And for example, putting together these two information can basically validate that the activation of this pathway would result in the same inhibition of the observed effectors. And by that way, you could basically puzzle together the credibility of the model that along this pathway and along this causality, you would have a good predictivity towards the observed outcome. Now, the authors also put in a more theoretical opportunity for us to consider later, and then you could switch actually to the next slide, which is this cross-pathway validation. And as I said, we have often in mind to create QSP models that are platforms that can predict across modalities, across pathways, across compounds. And what we will then need, right, is basically evidence and validation data that can validate that when we switch context, right, that we really can collect different validation evidence from different pieces. To make that a little bit more illustrative, right, if we have this example of the pro-inflammatory pathway, if we then can prove, right, against data that modulates, for example, the regulatory response, right, and still it's correct, that means that overall description of the immune system has been kind of correct. Unfortunately, at the moment, there are very few examples that attempt this kind of cross-pathway validation, which I guess goes a little bit into the line of thinking, as Armin pointed out, that we would need to come to basically a package, right, of pre-qualifying some sort of starting point and some sort of platform, which we can base our development and our model-based analysis upon, and then basically reduce the amount of qualification or pre-validation that's necessary then when we submit this to regulators. Also in this study, the existence and the usefulness of virtual populations has been pointed out, but again, has been not the focus, right, of this paper, which lets me to continue to the next topic that I have. And then, thank you. Exactly. So, virtual populations are becoming an increasingly important and essential part of QSP modeling, and nowadays you cannot really dissect QSP modeling from virtual populations that go alongside with them. So just to give you a quick idea of what virtual populations are a means to basically introduce between patient variability in your model, very often by statistical modeling in conjunction, right, with samples from the statistical model, or with exact counterparts of individual patients. And this becomes actually increasingly important the more right you go in the life cycle, right, of a drug. So starting from phase 2, you get interested in the ranges of behaviors that you try to see, and then once you move basically into the post-approval setting, you actually start be interested in the individual patient and what could be like an individual optimized dosing, or what's your individual benefit risk profile for that exact patient. And that kind of reflects this duality in virtual populations that are used in QSP. QSP current virtual populations, they kind of aspire to represent both the laws, right, of how generally variability or covariability can be described mathematically. And second, it really wants to capture also realizations within that variability. So individual, let's say, points or individual virtual patients within this cloud that you can describe mathematically. So this duality is kind of the leading feature of virtual populations in QSP. If you go to the next slide. So where do we stand? There's been recently a workshop that I would like to quickly report that's been sponsored by the ISOP, by the Special Interest Group of QSP, which basically aimed at finding, looking into use cases and summarizing the landscape and finding some, let's say, consensus evaluation criteria, especially for virtual populations, with the aim to better guide regulatory submissions and establishing clear applicability assessment guidelines. There's been a number of different speakers here, and for example, a relatively large, let's say, landmark analysis from Steve Chang, and also other use cases, right, from different pharma companies that really touched upon, let's say, regulatory grade applications of QSP modeling across different types of modalities. And what I would like to give you just a quick download of what aspects have been discussed, so that we can wrap up some best practices. Could you go to the next slide? Yes. So setting the stage. First, we consider there's really a growing number of successful applications. There's been a growing number of use cases where QSP models and the virtual populations have impacted regulatory impact, or have had regulatory impact, which really says that there's now really a critical time for us as a community to define what's a credible virtual population. What crystallizes out is that there's really, when we referred that before, diversity of different applications, workflows differ from use case to use case, and also the data that's been used for the validation is very different. So really we are in a context-dependent assessment, and we need better tools actually to aid with that context-dependent assessment, even beyond this model risk and model impact type of consideration. And there's been an earlier, let's say, paper on defining, a multifactorial grid, right, what that context might look like. And just to give you basically a sneak peek in what this group has suggested, so this has been taken from Steve Chang's presentation. This would be further evolution, right, of this multifactorial, multidimensional grid. And where here for each application, you would really go through basically what development phase am I in? Is this a clinical development? Am I more interested in safety versus efficacy? What types of data am I mainly recapitulating with my model? Do I have sparse data, rich data? Are there other clinical trials that I can potentially use and serve for my validation exercise? Are there potentially other therapies, right, for cross pathway or within pathway validation, right? And what type of validation do I actually target, right? As I said before, would it be within target or within pathway or even cross pathway? So this is really indicating where basically the journey goes. So multifactorial, multidimensional assessment or navigation basically of factors that can lead to my best validation strategy. So what also crystallizes out is, due to the diversity of different applications, not every virtual population is the same. We have quite a number of different virtual populations, and also virtual populations do not serve only as a tool to generate intra-patient variability. They sometimes just help us with a calibration, or they actually represent uncertainty rather than variability. And that's why we actually need to know how we generated that virtual population and what it actually represents. So if you can go to the next slide. I'll just summarize what based on the use cases has been discussed and what the kind of landscape about techniques are to come to a virtual population. You typically start, right, with one point and parameter space, one reference patient, and typically you build a virtual population by some crude assumption okay, there might be a statistical model around this, and there might be some covariability that I can describe with some multivariate statistical model. And then you start actually considering, okay, from this, let's say, broad blob of variability that I create hypothetically, what are actually margins that seem plausible from a biological perspective. And you start sampling and filtering and reweighting these patients so that they comply actually with biology. This is called a plausible virtual population. And then there's another step very often where you further filter, further reweight, right, from that cloud of points to actually come to a virtual population that is in line with basically the statistical properties of your target data, of your target virtual population. Right, and there are two, let's say, target virtual populations that you can look for. The first one would be the traditional virtual population, where this cloud of points represents the overall statistics that you have. So you're rather interested in the shape of the cloud of these points rather than actually the individual patients. I mean, they should all comply, but they should comply like in a general meaning. And this allows you then, right, to have, for example, multiple of these points and have a larger sample size than what you have actually in reality. The alternative, let's say, goal would be that actually each of these points should really resemble a real counterpart in your clinical trial, and this would make us more digital twin type of virtual population. Now, instead of this, expansion, filtering, and reweighting, you can also directly, right, optimize, for example, with global optimization, each of those points, right, to one of these points, and go directly, right, from your model, right, to a virtual patient. This is an alternative strategy for the virtual patient generation if you do that n times for every real patient that you have. And then you kind of also end up in a digital twin virtual population. And here you have also the opportunity, right, to reconstruct, the laws by looking into basically the sample of virtual patient and try to infer, right, how could the law actually look like, right, that would resemble this series of points. And then you can actually also combine and circle these type of methodologies to actually navigate and work with variability and actually patient samples. Next slide, please. And I try to make that short. So given the diversity of applications and the use cases that have been presented, there is, some elements of good practice that stand out, especially for virtual populations, it is very important to check the calibration goodness of fit. This is the starting point and considered to be the most essential element, because already capturing the calibration data with the virtual population is, I guess, one of the most important aspects. The data strategy, so the keep out or hold out strategy, or the prospective external validation strategy, is of course retained as one of the most important elements as well. And as a third point, right, we as a community still use quite a lot of diagnostic plots and visual predictive checks actually to check then the goodness of fit and also the prediction quality, right? And this is both for the ranges that we predict, but also for the temporal kinetics, so the temporal dynamics of the system with its basically prediction bands, and how landmarks or absolute values of your outcomes are actually distributed when considering that virtual population. And of course, right, predicted observed plots and other diagnostic plots that are used in other techniques like in pharmacometrics that are likewise important. Despite this clear bias still currently on diagnostic plots and visual checks, there's lots of statistical metrics that are increasingly used. For example, tests on the equivalence of distributions and other statistical metrics. So there's quite some traction on using statistical metrics. And in line, right, with what I mentioned before from Brackmann, uncertainty assessment really becomes or is an issue. And is an important element of the VPOP assessment, right? So we can display and we can use the virtual population as a tool to actually assess uncertainty. And you see, for example, here on the right-hand side, the approach of alternative virtual populations to check whether if we have a slightly different virtual population, does this really impact my prediction or not. So these are really important elements that kind of synthesize out. And I think I have one last slide. Yes, which is, this is really not the final point. This is just an ongoing discussion. So you can still contribute, right, to a survey that's being conducted at the moment, which is to capture what to do and what not to do for VPOP assessments, and how will digital twins fit within the assessment landscape, and what does the future hold. So with that, I think, we try to capture quite a lot of the journey of QSP and virtual populations, and that I give back to Dan.
Yeah, thanks very much, Alex. As I said, lots of food for thought there. Thank you very much. Right, in the interest of time, I think we will wrap up this part. Maybe, Alex, I'll give you maybe which bullet point from your industry outlook would you highlight, and then we'll go to Raj for one. So keep brief summary outlook.
Yes, maybe. I think what's interesting is that, of course, we get a lot of visibility from successes, right? From success stories, but less so on what does not work. And I think it would be, from an industry perspective, right, also share and discuss the failures and the learnings from examples that did not work, because what you see around you, right, is only the success stories. And I think that's a very valuable discussion that we could have and could foster a little bit more and have a little bit more.
That's great. Thanks, Rajat.
Yeah, thank you, Dan. I would probably pick the second bullet point, which is the optimal deployment of QSP across different phases of drug development. For instance, in the context of validation that Alex and you spoke about, with discovery, for example, it's more of an internal decision where we might have a list of candidates and we want to pick the most optimal ones for the next cycle. And therefore, we might be interested in something as crude as rank ordering these candidates so that we pick the top ones. Whereas when we actually get into the clinical part of it and the regulatory part of it, safety almost always becomes the overriding concern. And as you mentioned, regulatory agencies are very successful at keeping us safe, because vaccines go into babies, they go into adults, they go into everything in between. So contextualizing model risk and the influence of the model, depending on the context of use, is, I think, the most important thing.
Right, thanks. So we'll come next to regulatory perspective. So Sue, again, interested in time, maybe one or two key points you'd like to address here.
Yeah, I think most of them speak for themselves, actually, and have been covered already by some of the other speakers. So maybe if I had to pick one, I would say we do encourage the use of these models in submissions, particularly the QSP models that give us that mechanistic understanding, and look out for any guidelines that are out for comments. And if you feel modelling could be useful, please do comment on that.
Perfect. Thank you very much.
So thank you to the panelists. I put together a brief summary slide of a few key points. We did speak today about a specific context of use that had kind of low to moderate risk, and a question, I guess, going forward would be, okay, if we want to take that to other contexts of use where the risk is higher, are we going to be talking about other credibility factors? Although I guess we need to be starting somewhere and building consensus, which is easier at the lower risk levels. We heard already about the M15 and applicability of the ASME 40. And we also heard about the QSP models around what correct biology and model trajectories generated for the correct reasons. Okay, so with that, I think that's all we have. So thank you everyone for attending today.
Thank you, Dan, and everyone who attended. Take care. Bye.
Thank you. Bye-bye. Thanks a lot.
Bye.